Okay, welcome everybody to this month's installment of the Chalmers AI for Science seminar series. This is a seminar series where we host young, excellent researchers from across the world to present research about AI and their interactions with the natural sciences typically. And so today we have Anne-Florence Bitbol from the EPFL in Lausanne. Anne-Florence did her undergraduate studies in Lyon and then moved to Paris to do a master's. And after that also stayed in Paris for a PhD. And then since, and before joining EPFL in 2020, she was a CNRS researcher, permanently employed by the French state, but then she decided to take up on a tenure track position, and she's been there since then. And so we're very happy to see what she has to present about today about her work on protein language models. Welcome.
Well, thank you very much. I would like to thank the organizers, Simon and Rocio, for this great invitation. It's really very nice for me to, to get to know you and to have this opportunity to discuss and present my work. Okay, so I'm going to tell you about some recent work in my group. And just before I start, I will put a bit of context. And so in my group, we are interested in evolution at the scale of proteins and protein sequences on the one hand, but also at the scale of microbial populations on the other hand. Today, I will really focus on the protein parts and specifically on the impact of machine learning on this side of our research. So I will tell you how we can use protein language models for various applications, such as predicting which proteins interact together and generating new protein sequences. Okay, so this is an outline of the talk. I will start by introducing a little bit protein language models, and in particular, those which are based on multiple sequence alignments. And then I will tell you about some various aspects of our work. Okay, so let me start with this introduction. And I would like to start with the data. By the way, I should have said it from the beginning, but I'm happy to take questions. So please feel free to ask me if there is something which is unclear. All right. So this plot is showing you the number of sequences which are available on one given public database, which is called the European Nucleotide Archive. And as you can see, this number of sequences is growing extremely fast in time. And in fact, this curve is quasi-exponential. So there is some redundancy in this data in the sense that there are some sequences which are very similar, maybe because they come from very close organisms, like two different strains of the same bacteria. But nevertheless, sequences are not double counted in the sense that, like usually if it's really the same, you don't count in twice. So there is really a lot of data, more than 10 to the 9 sequences. And this all comes from the advance of genome sequencing, which is getting more and more scalable and easier. And this data is a great opportunity for methods which are based on various computational approaches, and in particular machine learning, to learn about proteins. Something I should also say is that in some sense, we can view this mainly as unlabeled data, in the sense that we have the sequences, but we don't really know the function or the structure from experiments, right? Like determining the structure of a protein or the function of a protein is much more costly in terms of experiments than determining the sequence of a protein or of a genome. So many computational approaches are working on extracting information from these proteins. And one more thing I would like to say about this type of data is that we have this huge ensemble of sequences that exist in nature, but it can actually be organized into some families of proteins. So what is a protein family? It's a set of proteins which have similar biological functions and similar 3D structure. So here you can see an example of a 3D structure taken by a protein. It's like the way the protein spontaneously folds. And maybe I should say that, in fact, like just the mere fact that a protein folds is not something which is obvious, in the sense that if you take a random sequence of amino acids, then generally it absolutely doesn't fold in a well-defined 3D structure. But the proteins which exist in living organisms have been selected by natural selection to perform given biological functions and to fold in a well-defined way. And so these are the ones that we see in the data that is sequenced from living organisms. Okay, so I was talking about protein families. So here you have to imagine that we collect some proteins which fall into this same structure and have a similar function. And they are similar also from the point of view of their sequences, at least if they are similar enough. So in fact, these proteins have descended from the same ancestral protein and they have evolved. And in this data structure here, you can imagine that the first row, for instance, is the human hemoglobin, and the second one is the chimpanzee hemoglobin. And so they are very similar, but there are a few differences. And the sequences, if they are similar enough, can be aligned, which means that each row is a sequence, but also each column is a given site that has a given position in the 3D structure and a given functional role. Here, my example is a toy example, but like, okay. It's just a small example to show you the type of data. And the important thing is that these proteins really share function and structure and ancestry. And there has been a lot of work trying to start from this kind of data structures, these multiple sequence alignments of homologous proteins, in order to infer protein structure, protein interactions, and protein function. And one key element which allows to do this is the fact that there are some constraints due to the need to maintain structure and function. So here I'm showing you a simplified example to give you the idea of these constraints. And here you can see the amino acid chain which forms the protein. And there are two amino acids which are in color here, and they are close to one another in the 3D structure. And this is something which is important to define the 3D structure of the protein. These are two amino acids that need to be complementary in terms of physics and of chemistry to maintain the 3D structure. But during the course of evolution, you may have a mutation that happened at the level of the sequence, and it may affect one of these important amino acids for the structure. And in this case, okay, so here is just a change of color. So this amino acid is no longer complementary in terms of physics and chemistry. And because of this, the structure of the protein and also its function are impaired. This is bad for the organism which carries this protein because presumably it's no longer able to perform its function well. And so this state is going to be eliminated by natural selection. However, you could have another mutation that happened at this other amino acid and that restores this complementarity, right? So because of this, when you observe the sequences which have survived natural selection here to maintain this structure, you will observe this correlation between the amino acid state at these two sites which are in contact. Yes, question.
How are post-translational modifications handled at the sequence level, or is this something that like you don't consider at this stage?
Yeah, that's a great question. So I mean, here we don't consider them, but it's true that they can have an important impact. Here we rather focus on the data that can be obtained directly from sequencing, and so that's kind of limited in this respect.
Okay, so there's no sequence representation for that?
You could add it, but for now, like yeah. Sorry. Maybe something I should say also about this example is that it's a very simplified example. This example of the compensatory mutation. In fact, like I told you, like one mutation really impairs the structure. Usually, I mean, most mutations will slightly impair, but not fully. So things are a bit more subtle, but that's the qualitative idea, that you need to have amino acids that co-evolve in order to preserve structure and function. So this kind of signal has been studied for a while, and the idea is rather to take multiple sequence alignments and to try to look at these correlations and to infer properties from that. And one approach which has been very successful is the POTS model, which aims to construct a priority distribution of observing a given sequence in the family that we are focusing on. And the priority of a given sequence is written in this way. You have the form here. And in fact, it's a Boltzmann distribution associated to a given Hamiltonian, a given energy function, where you have terms which involve only one site. So I here is one site, and you have a sum over I. So each of these terms, hi, involves one site. And then you have also terms which involve two sites, ij, that involve, this kind of like one, I might be this one and j might be this one, for instance, and summed over all the pairs. So these terms here will include the interactions between these two sites. So this model has a nice form in terms of, being a Boltzmann distribution. And it's called post model because the Hamiltonian that is inside is the one of a post model. A post model is just a generalization of an Ising model to more states. Here in the case of the protein, because there are 20 natural amino acids at each site, and we may have one of these 20 amino acids. Okay, so that's one approach which has been quite successful, and the strategy here was to infer this post model from the data. So you have the data and you want to find the model which fits the data. And the way it's done is that you want to find disparity distribution such that it matches the frequencies observed at each site. So the marginals are the same, but also the two-body marginals, which like you take two sites and you marginalize over all the rest. This should also agree. So this model can be constructed, in fact, by maximizing the entropy among the models which match these constraints. So that's one way to find this form. Okay, and so these models have been very useful for instance, to predict contacts from sequences and so on. But now there is a whole new class of models, which is called protein language models, and AlphaFold2's evoformer, which is part of the supervised structure prediction method of AlphaFold, and that actually just got the Nobel Prize, is something which uses a protein language model based on multiple sequence alignments. So AlphaFold is, comprises also a structural module, but this part, the sequence-based part, is in this category of models. Another one, which I will tell you about more, is called MSA transformer. It's a self-supervised model in the sense that it was trained only on sequences, not on structures. AlphaFold was also trained on structures, the experimentally determined structures, right? And these models use the attention mechanism and the transformer architecture, which come from natural language processing. And so, because AlphaFold is in the news again, I put this slide about AlphaFold2, which is the model that actually, won the structure prediction contest in 2020, with a score which is such that the protein folding problem could be claimed to be solved, right? So that was the big advance. And AlphaFold2 has a model which has many different blocks. It's a quite complex deep learning architecture, but it has this block which starts from sequences and from multiple sequence alignments in particular, that is called the evoformer, which I circled in red here. And this evoformer is a protein language model trained on multiple sequence alignments. Then it's coupled to a structure module, which actually, here I put the version of AlphaFold2. I would like to note that the structure module has completely changed in AlphaFold3, but the part on the sequences has changed less. Okay, so that was, kind of motivation to look at these kinds of models. But now let me tell you a bit more about how these models are trained. And for this, I would like to say just one word from natural language processing, which is the objective with which these models are trained. So these models are trained with the masked language modeling objective, which is the objective of filling in a masked word using the surrounding context. And so here I gave you just an example of a sentence. The man went to the and bought a of milk. So yeah, there are two masked words here. And you hope that the model, after seeing a big corpus of text and being trained properly, is going to learn some sensible guesses here, such as the man went to the store and bought a liter of milk. For this, the model is trained to minimize the pseudo-likelihood loss, which includes the probability here of having the right words given the context. So it's really focusing on the context to find which word to suggest. This is for language. But in proteins, this can be adapted quite directly. And here I'm going to show you how it was adapted in MSA transformer, which is a self-supervised protein language model trained on multiple sequence alignments. And I'd like to mention right away that there are also other protein language models which are trained on non-aligned sequences, like a big set of sequences which are not organized into these alignments. But I'm going to focus on those which are trained on alignments. Okay, so the data structure we have is a bit different from sentences. It's rectangular matrices where rows are sequences and columns are sites. But you can consider the amino acids as words. Say that you mask some of the amino acids and you want to predict them using the surrounding context. So that's what was done in MSA transformer, which was introduced by Rao and colleagues in 2021. And the loss, which was used to train the model, is a pseudo likelihood loss, which is quite similar to the one I showed you before, except that now it's amino acids instead of words. Okay, so that's the idea. Yes.
Complex there, like a 2D kind of complex then, or?
That's right. So you're right. I mean, like here, the data structure that you start with is really a like 2D matrix, rectangular matrix. So indeed, you need to calculate attention coefficients on two different dimensions. I will actually come back to this just after. Other questions? All good? Okay. All right. So the architecture of MSA transformer is really a transformer architecture. It's in fact based on the BERT-based architecture with 12 layers and 12 heads of attention. The total number of parameters is not huge. It's 100 millions, and the Evo former of AlphaFold2 is similar in size, 91 millions. And there is one thing, though, which is a bit different from language, which is this rectangular structure of the data. And so the attention had to be adapted for this data structure. And for this, we need to think about what is the relevant context to predict a given multiple sequence alignment. Sorry, a given amino acid in a multiple sequence alignment. So let's think about this. Here you have this rectangular matrix, which is your multiple sequence alignment. And you want to predict this masked amino acid here, which is in red. And for this, what is going to be the relevant context? Well, the first thing that is going to be relevant is the column that the amino acid is in. Why? Because there are some columns which are extremely well conserved across evolution because they have a very important function. And so basically, if you have a mutation at this site, it's super bad for the protein, right? So the column is going to be very important, and that's column attention. And then the column is not enough. In particular, we know that there are these correlations in amino acid usage. So just having an independent site model is not sufficient to describe proteins. Because of this, you also need to have the context of the row. And within the row, you will probably need to give more attention to some specific spots, right? But anyway, the key point is this, that you start with this axial attention framework where you have column attention and row attention. And then here, because we know already about coevolution, in fact, it is useful to impose that the row attention should be similar for all rows. And the idea here is that if you want to predict this amino acid and you find out that it's nice to give a large weight to this part of the context, then usually if you want to also predict this amino acid, it's going to be useful to put a big weight on that one, which is in the same column. That's because of the importance of these correlations between sites, between columns.
So is this essentially like a low rank approximation of the full attention? Because if you would have a dense attention, you would have basically the whole rectangular matrix would have
Yes.
to tend to be like an alpha of a few attentions.
Okay, so you're completely right to say that in principle you would want to put attention on the full rectangle, right? Yes. However, it's quite heavy to do this, and one of the motivations to focus on rows and columns is that it's lighter. Mm-hmm. And then, like, I mean, okay, the low rank picture, I agree that qualitatively it's a good one, but I'm not sure if it's exactly the, yeah. Question?
Can you shuffle around the rows as you wish?
Ah, great question. Okay, so you didn't ask the question for the columns because you know that it's important, right? Yes. So in fact, there is positional encoding both for the rows and for the columns in MSA transformer. But you are right to say that shuffling the rows is far less problematic than shuffling the columns. And in fact, if you were to shuffle the rows, you would still get some attention coefficients, which would be different, but like they would be, I mean, a form of permutation of what you would get before. That's the way it works in the end.
So then do you always follow a specific ordering for the rows, like according to species? Or...
Ah, yes. Well, I mean, okay, you can feed the alignment in the order you want. And again, like it's not going to matter in the sense that the attention coefficients which need to be big in this column attention will be. But if you want to study phylogeny and so on, it's relevant to do this ordering that you were suggesting. Also, maybe one thing which I didn't say here for the sake of time is that when MSA transformer was trained, they also had to figure out how to subsample alignments. And it turns out that it's better to subsample them by keeping a quite high diversity instead of focusing on very similar sequences, so that you learn more information. Okay, so here, this is one more point about architecture. It's like, okay, again, we have column attention and row attention. And the point is that in MSA transformer, the row attention was tied in order to impose that for different rows, it would be the same. And then you have row attention and column attention, which are stacked, and there is a feed-forward layer at each stage. Okay, and this was, this model was trained on a very large data set of many multiple sequence alignment, 26 million, in fact, which correspond to a vast overview of the proteins families that exist. So it's really a very wide model, which has seen a lot of protein families. Okay, so now let me show you a little bit what MSA transformer captures. For this, I'm showing you first a plot which comes from the original MSA transformer paper. It's showing you the ability of MSA transformer at capturing structural contacts in 3D. So which amino acids are close to one another in the structure. And remember that the model was trained only on sequences, right? So here, the success of this contact prediction is shown in percentage versus the depth of the multiple sequence alignments, which is the number of sequences you have in the data set, right? Number of homologs that actually exist. And it's always easier to make predictions when you have deeper multiple sequence alignments, because you can learn the statistics more easily. And that's something that has been true also for POTS models. So here, the green curve corresponds to POTS models, the ones that have this formula, which we saw before. In POTS models, you have to build one model per family, because it's learning the coevolution within, like between sites in a given family. And it's known that POTS models perform well when there are enough examples, and far less well when you don't have enough examples to properly learn the correlations between sites. So that's why the green curve has this strong increasing trend. You can see that the curve, which is coming from MSA transformer, the blue one, also has an increasing trend, but that is clearly above, especially in the regime where the depth of the multiple sequence alignment is small. So MSA transformer, again, because it was trained on many protein family, shares parameters across these families, and presumably this helps it to address the difficult cases of the small families. Something I didn't explicitly say is that this contact prediction here is obtained from the row attentions, right? The row attentions are the coefficients which, like we're here, like the idea is that, these are the coefficients that tell you this part of the context is very important to predict this amino acid. And expectedly, in some sense, from the point of view of coevolution, this captures the sites which co-evolve and which are in contact.
So you require the contact to be in one direction or both directions? Yeah, to infer contact.
So, okay, so the way you do it here is that, okay, you have attention matrices, which are calculated by the model on your multiple sequence alignment. And in fact, okay, there are many of them because there are 12 heads and 12 layers. So in fact, if you look at them, you already see the contact map emerging in many of them, right? But there is a way to improve this, which is to build a regression that, you find a combination of these matrices, which will predict the structure, and you can train it in a lightly supervised way on a few structures. In practice, in the paper, they used 20 structures, and that was sufficient to find a regression that was universal across. Yeah. So that's the way it's done. It's like really by taking the attention matrices. Okay, so that's, okay. So far, we saw what information there is in the row attentions. It's the structural contacts. Okay, at least, there could be more, but at least there is the information about the structural contacts. Something that we asked in my group is what kind of information there is in the column attentions. And we already had this discussion that, the rows can probably be permuted and so on. So to address this, okay, we had this idea that when we look at multiple sequence alignments, the columns are correlated due to contacts. Some columns are in this category at least. But the rows can be similar due to phylogeny. So we wondered if the column attentions were going to inform us about sequence similarity and phylogeny. A qualitative way to see this is that, you know, if you have two sequences which are very similar, it makes sense to give it a lot of attention because you can again copy a little bit from it, right? So to address this, we built a logistic model of the column attention matrices. So similar to before, except that we are working on column attentions instead of row attentions. And we tasked this regression to predict the matrix of pairwise Hamming distances in multiple sequence alignments. And we did this for a given training set of multiple sequence alignments, and we tested on other multiple sequence alignments. And we found that the predictions using this regression are pretty good, as you can see on these examples here. And it means that a simple combination of the column attention coefficients is encoding the Hamming distance between the proteins. So it captures the similarity between the proteins and these phylogenetic relationships.
So how much is this just reproducing the data that you get basically from the multiple sequence alignment? Because multiple sequence alignment is basically assuming some model of evolution, right? So you're inferring them while you're doing the MSA. And so how much are you just regurgitating the data pre-processing?
I mean, you know, I think the whole question of inferring phylogeny from this model is super interesting. This finding here is kind of simple in the sense that we just looked at Hamming distances. Of course, the Hamming distances, as you pointed out, depend on how you align on the proteins in the first place, right? But like here, the question is really, is the model capturing, like learning to calculate these Hamming distances in some sense, right? Is there an
sort of like a sequence metric?
Exactly, that's it. And so I mean, your question about like whether the model is in some sense biased by the type of alignments it has seen and so on, I think it's a very interesting one, but I don't think it's really addressed by this. Here we are just seeing that the model is computing or learning the distances. Maybe something I should say here is that we can see some block matrices. And the reason why there are these blocks is because the sequences here were ordered by phylogenetic order, meaning that close sequences were close to one another, and here the light colors mean that the Hamming distance is small. So yeah, that's kind of going back to the question. And then we are comparing here the ground truth and the predictions. Okay, so that was finding which I didn't want to discuss too much. Now I would like to move on to how we can use MSA transformer in order to infer which proteins interact together among the paralogs of two protein families. So our problem is the following. We have two protein families, A and B, which are known to interact together, for instance, in one model organism, right? You know that one A interacts with one B, but then you have many organisms and you have a big data set of proteins of family A, another one of proteins of family B, and you would like to know which A interacts with which B within each species. So that's a question that arises because in a given genome, there are often paralogs of the same, which come from the same family. And your question is, does A1 interact with B1 or A1 with B2, etc.? We assume here that the interactions are specific and that they only have one partner. And for this, we can actually use coevolution. So you see here a figure which is quite reminiscent to one I already showed you, except that here you have two chains, A and B, right? And the chains A and B interact together through amino acids, which need to be complementary in terms of physics and of chemistry too. And because of this, if you had the correct pairings, you would observe these types of correlations, right? However, if you don't have the right pairings, you would presumably observe lower correlations, right? So our problem is going to become to find the pairings which maximize this coevolution signal and the correlations. So this is a problem which has been treated in various ways, and I'm not going to to tell you the details about this, but the rough idea is that you want to maximize the coevolution signal. And we proposed recently a new method which uses MSA transformer in order to address this problem. So to do this, we take some data sets where we know the answers, we know which A interacts with which B, and the good news is that there are such data sets because in prokaryotes, if you consider two families which are known to interact together and which have many paralogs, then usually the correct partners are encoded in the same operon. So you can just take the ones which are in the same operand and they are the correct answer. Okay, so let's say this is a kind of ground truth data. So we have this paired data set where we know which A goes with which B. And to begin with, to see if MSA transformer can capture this kind of signal, we start with such a paired alignment and we feed it as input to MSA transformer. Remember that MSA transformer was trained on single family multiple sequence alignment. So in principle, it has not seen this kind of data. And we first asked whether the coevolution between the chains is actually captured. To address this, we looked at the contact prediction. And here I'm showing you two contact prediction that we did. The first one is in the case where the A's and the B's are paired in the correct way. So these are the right pairs. And here you can see a contact map. Essentially, it's dark blue if there is a contact and light blue if there is no contact. This is the full matrix. And here, these are the details for the first square, which is one protein, the A. This other square is the B. And the rectangular part here is the interprotein part. So the one we are interested in, the one which is between the A and the B. So it's this one, this rectangle. And you can see here that there is a V of blue things, right, in this interacting part, which actually corresponds quite well to the experimental ground truth from X-ray crystallography, which I'm not showing here, sorry, but like, you know, like this V is very characteristic. So it's captured very well in the case where the A's and the B's are paired correctly. But now, what happens if we shuffle the A's and the B's? Well, here is the answer. We find that this V is lost, right? Here in this rectangle, you have only white. There is no predicted contact from MSA transformer. So this means that the coevolution between the A's and the B's is captured in the sense that the contacts are predicted if the pairing is correct, but not if the pairing is incorrect. So that's the first sign that MSA transformer could be used to address the problem of pairing. Then the second question we asked was whether the loss, the training loss of MSA transformer, the masked language modeling loss, actually depend on the pairing of the A's and the B's. And so here I'm just reminding you of the formula of the loss, but here are some plots of this loss versus the number of correct pairs in a data set. So here, I mean, essentially it varies from 0 to 50 because our data set had 50 sequences, right? So essentially, when it's 0, you have no correct pairs. When it's 50, all the pairs are correct. And what you can see is that depending on the masking probability, which we varied here, you can have some nice decreasing trends. So here you can see decreasing trends. These decreasing trends mean that the loss is lower when the pairs are correct, right? So that's a good sign. It means that we can now formulate our problem of pairing as a problem of optimization that we want to to minimize the loss. Okay, so that's the way we formulated it. So in each species, you will want to find the permutations of the B's with respect to the A's. So, you know, for one partner with respect to the others, that minimizes the masked language modeling loss. And optimizing over permutation is something which looks really difficult in the sense that permutations are very discrete, right? But fortunately, there is a whole branch of machine learning which aims at making discrete problems differentiable and continuous, right? So we use the synchrone operator, which actually allows to approximate permutation matrices by continuous ones. And so the problem becomes differentiable thanks to this trick, and it can be solved using gradient methods. So that's what we did. And here I'm showing you some results that we obtained. And these results are obtained on one of the benchmark cases I mentioned before. So a case where we know the answer, we can just compare to the ground truth. And so this score here is telling us what fraction of the predictions are correct. And here we are looking at a case which is actually very difficult for all traditional coevolution methods in the sense that it only has 50 sequences. So very few, and that's the kind of regime where traditional coevolution does not work well. As you can see actually in the baselines, these dashed lines, dashed black lines are the traditional methods, and they have only less than 20% success, a bit more than the chance expectation, which would be 9%, but like not much more. And here the three colored lines show the performance of our model, which is based on using MSA transformer and the masked language modeling loss. And you can see here the performance over the whole data set. And in red, you also have the performance over the 10% pairs, which were given the top confidence by our model. So what you can see is that we have better performance than traditional methods, which is, okay, really far from perfect, but also confidence is a good metric of whether the pairs are correct. Okay, so that's just one example. But then we can also ask other things. For instance, here, we started our prediction problem without giving any examples of known pairs. And maybe giving some examples can help. So if we give some examples, we are going to call them positive examples of correct pairs. We can give them as context in the alignment and just not mask them when we make the predictions. And this can actually be quite useful, as you can see here. This is the success of pairing versus the number of positive examples, and you see that it increases with the number of positive examples. Okay, and then another thing is, so far I showed you results from a very small alignment. Is it going to actually improve if we have larger alignments? And you can see that yes, if we make the alignment larger, we have better performance. This one is just another example on another benchmark case, just to show that it also works there. But now, so far, I only told you about benchmark cases where we already know the answers. And so that's not super exciting, right? But we also looked at an application of our method to eukaryotic complexes. And in eukaryotes, we don't really know the answers from genome proximity because there is not the nice operand structure that there is in bacteria, right? So it turns out that there the problem is far less easy to solve. And actually solving it can be useful potentially to predict the structure of multimers. In the sense that AlphaFold multimer, which is the version of AlphaFold that predicts the structure of complexes, actually uses paired multiple sequence alignments as inputs. In fact, all AlphaFold methods use multiple sequence alignment. When you give them a sequence as input, they actually look for homologs in databases and build multiple sequence alignments. But if you want to predict the structure of a complex, in principle, you need the alignment to be paired. And it was shown in these two papers that the pairing actually matters for the prediction. So we looked at some complexes which are not in the training set of AlphaFold multimer and for which AlphaFold multimer was not doing very well. And we found that in some cases, we have some improvements over the default AlphaFold multimer pairing strategy. In the sense that here we are showing the DocQ score, which is a score of quality of complex prediction, so a complex structure prediction. And in this example and that one, you can see that the performance is much better in blue when we use our methods diffpalm instead of the default AlphaFold multimer pairing strategy. So let's look at these two cases in more detail. Here you have the structure of these complexes. So the light colors are the ground truth from the protein data bank, the experimental ground truth. And the bright colors are the predictions. So here, this is the prediction from the default AlphaFold multimer version. You can see that the blue chain is not placed correctly, right? And when we use the alignment for diffpalm, in this case, we restore the placement of the blue chain. And it's a similar situation for this other one. Of course, it would be necessary to assess this on a far larger data set, but in some cases, it seems that it's useful. And it's also a sign that the pairing is probably relevant. Okay, so that was for the pairing. I'm going to be quick about the rest because I see that time is flying. But I'm happy to take questions about the pairing if you have questions. Yes?
Was there something in common, like the structures that the diffpalm was able to correctly predict and not AlphaFold multimer?
Yes.
Was there something that distinguished them?
Well, yeah, that's a great question. We wondered about this a lot, and it's a bit difficult to, like, because the number of sequences we were able to test, structure we were able to test is not huge. So it's a bit difficult to address it, but there is one thing which we saw, for instance, here, this one, 6PNQ is a case where the number of sequences that can be paired by pure orthology is not very large. So here, actually using our pairing method could give it a boost. But yeah, I mean, I think it's going to depend on the size of the multiple sequence alignments, how much data there is, how bad it is to make a kind of more naive pairing. This will depend on like, yeah, the number of sequences, the number of sequences per species and stuff like that. Maybe just to make one point clear, we are still using AlphaFold multimass, just that the input is different. Yeah, the alignment are different. Okay, cool. So let me tell you a bit about how we generated sequences using MSA transformer. And here, the idea is that MSA transformer a priori is not a generative model, right? It was trained using the mass language modeling objective. However, we can exploit this objective in order to propose a generative method. So for that, we started with natural multiple sequence alignments, and we masked some amino acids. We asked MSA transformer to fill in these amino acids, which is what it's good at, right? And then we iterated by taking this new alignment as input, and again masking and filling. And we did this multiple times until some of the properties of the alignment converged. Then, once we have done this, there are a few questions. Are the sequences that we obtain new sequences or not? And are these sequences still in the same family of proteins as the initial one or not, right? So we aim to characterize these sequences. And for this, we compare them to natural ones and also to sequences generated by POTS models, because the POTS model has been shown to be generative also by experiments in this paper by Ross and colleagues. So that's the thing that we are going to compare. And here I'm showing you some scores, which are Hammer scores. So the Hammer score is a homology score. It's a score that would be used to fish some homologs in a database. So if the score is high, it means that it's considered as a good homolog of the natural family we started with, right? So this is the Hammer score, and it's shown versus the Hamming distance to the closest natural sequence, which is our worry of overfitting, right? If this distance is too small, it means that essentially we are outputting sequences which have been seen in the training set, right? So the blue cloud here is showing data which was generated using our method based on MSA transformer. And you can see that, first of all, there are some sequences which have Hamming distance Hamming distance to the closest natural sequence that are not too small. That's a good thing. We are not overfitting. And moreover, we also see that the Hammer scores are pretty good, and in fact, slightly higher than those of the natural sequences. The slightly higher is maybe a worry in the sense that the model is a bit idealizing these sequences, but at least they would be determined as good homologs using this criterion. Okay, so this is another score, which is the score from the coevolution POTS model. And you can see here that the proteins that we generate are also having quite good scores. So here the score is defined in such a way that higher is better too. But we also compare to the POTS model generated sequences, which are in yellow and in green here. The yellow ones are the ones which are generated at low temperature, so sampling in the energy minima of the POTS model. And these are the ones for which it's known that generation actually works in experiments. You can see here that from the point of view of Hammer scores, it's pretty similar. And from the point of view of statistical energy scores, the yellow are better, but that's because they are actually optimizing this score. Okay, so this was for one protein family. We looked at other ones. This is another one, and the results are quite similar. And then we looked more broadly, and we looked at 14 large families where POTS models can be trained reliably. And we found that here too, the Hammer scores and the statistical energy scores could be compared well between the natural and our generated sequences. But moreover, we looked at two other scores, which are on the bottom here. These are AlphaFold scores. So we took our synthetic sequences and we fed them to AlphaFold and asked for structure prediction. And the first one is giving you the AlphaFold confidence. And the second one is giving you how close the predicted structure is to the structure of a natural homolog, which was determined experimentally. And you see that this score here, the root mean square displacement, is actually quite small, but also comparable between the natural sequences and those generated by MSA transformer. It's as low or even sometimes lower for our generated sequences, which is a good sign that they would fold into the same structure as the natural family that we started with. So it looks like these new sequences are synthetic homologs of the family that we started with. But so far, we are not really doing better than the POTS model, which is also generative, right? But then we looked at the case of small protein families, which are not very well captured by POTS models, in the sense that the data, like the multiple sequence alignment size, is too small to learn the statistics well. And in this case, it turned out that our model was still performing quite well. So the blue things are still similar to the red ones. And in terms of distance to the closest homolog, here the POTS model with low temperature in yellow is failing, in the sense that it's outputting sequences it has seen in training. It's overfitting because of the small size of the multiple sequence alignment, so that's expected. While our method based on MSA transformer does not seem to have this problem. So it means that the method is actually quite promising in the case of small protein families. Yes.
Is there a way to include some transfer learning into the POTS model?
Sorry, to integrate what in the POTS model?
Basically, you are leveraging on the larger number of families that the transformer is able to see. So I was wondering if you can add some bias in the POTS model.
That's a great question. I mean, I think, I mean, look, so far, what I compare to is the regular POTS model. I agree that the question of whether we could make the POTS model more aware of other families or training a kind of generalized POTS model across family is a very interesting one. There are some modified POTS models which take into account some local secondary structure features, and they are quite interesting already. But yeah, so far it's like the improvement, I would not say it's that spectacular.
Some people would argue that the transformer is a generalized POTS model.
Yeah, well, okay, that's also a very good point, which is that the like, you know, there is some nice theoretical analysis of attention, and that, you know, like one attention head can be actually analyzed in terms of the POTS model. At least in some cases, if it's factored, I mean, like there are some conditions, but there is a link. However, I would say that MSA transformer, because it's a large model and it has seen many families, does have some properties which are different.
So your intuition is that it is using some higher order statistics.
Great question. I mean, so I didn't put it in this presentation, but we also wondered whether the higher order statistics were better. Maybe I have it in the supplement. Okay. If the higher order statistics, yeah, okay, it's here. If the higher order statistics were better captured or not by our model than by POTS model. And it turns out that in general, yes. So here, these curves are showing a score which is quantifying the ability to reproduce higher order statistics. I mean, statistics of different orders, right? So the blue scores are MSA transformer, our data generation method based on MSA transformer. The yellow and green are POTS models. And you can see that often the curves start somewhat similar, or the green is above, right? Often. These are low order statistics. So the POTS model is better at low order statistics, which makes sense because it's fitted, like the first two, 1 and 2 are fitted by definition, right? But then for higher order statistics, the method based on MSA transformers seems to often do better.
Okay, and in the case where it is failing already, is it because the family is particularly small, like in the bottom left corner?
Sorry, in the bottom left. Oh, you mean this one?
You see that the POTS models are failing even at.
Yes, you're right. Okay, here is. I don't remember exactly, but it may be because the family was a bit smaller. Like, okay, I think it was not very diverse. Yeah. All good, no? Okay, I think the time is a bit. Maybe I should kind of. Should I remove the third part or just say a few words? I don't know.
I think it's interesting to hear about this. If you can do a quick one.
Okay, I will do a quick one. Sorry, I guess I'm a bit over time. All right. So our the last part here is about whether we can actually get rid of multiple sequence alignments. So far in my talk, I told you we start from multiple sequence alignments because they contain homologous protein, they allow to see coevolution and so on. But there are also some protein language models which are trained without alignments on a corpus of sequences which are not aligned. And for instance, there is the ESM class. So this is work by Lin and colleagues, ESM2, okay? So here you can see that if you look at these ESM2 models, this is a figure from their paper, the prediction ability for contacts, so again, the same problem as before, can we predict contacts using this model, structural contacts, depends a lot on whether there are homologs available and how many there are. So here, the prediction ability is shown versus the number of parameters of the model. It's increasing. Larger models are better. This is very much in the, like the spirits of these models. But the different colors correspond to different numbers of homologs. Darker colors correspond to cases where there are actually many homologs available, right? And you can see that performance is much better if you have more homologs available, despite the fact that the model is not supposed to know about homology, right? So it means that the model is learning homology on its own, and that it's able to use this signal, right? And so this was analyzed, and for instance, there is one interesting paper that says that protein language models store evolutionary information in their parameters. Yeah. Okay, so multiple sequence alignments are useful. It seems that even models that don't use them capture the signal from homology. So we wanted to ask the following question, which is, can we exploit homology in an explicit way without using alignments? And the motivation for that is that alignments can be imperfect. Like, we never really know how to fundamentally align. So we proposed a method where we concatenate homologs instead of aligning them. And this is something that actually has been done in a model called POET also. But we did it in a different way in the sense that we didn't use a transformer or attention. And the motivation for that is that if we have these long sequences which are concatenated, it becomes a very long context, and it becomes difficult to actually make calculations of attention which are quadratic. And so we used a model which is called Mamba, which is a state space model. And the advantage of this model is that it can handle very long context. So in our case, it can handle hundreds of concatenated sequences. So we trained this model with an autoregressive generation objective. So it should be able to generate new proteins, and also a fill-in-the-middle objective, which means that it's able to also use context both on the left and on the right to fill in some piece of the sequence. So the hope is that it's going to combine the strengths of autoregressive modeling and mass language modeling. And then we tested this model. So far, this model is still a rather small model. It's a bit of a prototype. It has 100 million parameters. And we tested its ability at generating new sequences, very similarly to what we did for our method based on MSA transformer. So we want to generate within families, and we find that the results are rather promising in terms of finding structures which are consistent with the expected ones, and also homology scores which are quite good. I would say here that the perspective would be also to test the fill-in-the-middle objective by performing sequence infilling of an important piece of the protein that you want to modify, given the context. So if you have any ideas of specific case that we could test for this, I'm happy to. All right. Another thing that we tested is the ability of our model ProtMamba to predict mutational effects, so the effect of fitness of a mutation. And so far, it seems that the results are encouraging in the sense that on the protein gym benchmark, which is from this paper, we have results which are comparable to some state-of-the-art models. Some of them are actually much larger than our models. So it's encouraging. And another thing that we tested is the prediction of enzymatic activity on a specific data set of chorismate mutases. And it also gives some rather promising results. So we still have a lot of work to do on this ProtMamba, but really the spirit of it is to try to exploit homology without needing to align the sequences. Okay, so that's it. I would like just to conclude here and to say that protein language models trained on multiple sequence alignments capture contacts and phylogeny in their representation, which I think is quite remarkable. They also capture interchain coevolution, which can be used to pair interaction partners. We can use them to perform sequence generation. And now we are trying to also build models which are not using alignments. If you'd like to know more, we have a few references here. And I would like to end by thanking the people who worked with me on this project. And in particular, this work that I presented today is mainly work from my former postdoc, Umberto Lupo, my current postdoc, Cyril Malbranc, and my current PhD student, Damiano Sgarbosa. And I would like also to thank all my current group members. Some of them are working on the population scale, so I didn't really get to talk about their work, and the alumni. And also some colleagues for our discussion, especially about, like MSA transformer when we were starting to use this type of models. And I also thank my funding sources for generous funding. And I thank you all for your attention.
you, Anne-Florence. Do you have any questions in the room first? Yeah.
I have a question. So you make use of decades of protein research, generated this very nice database. But they also have found many other ways to analyze the data. And can you somehow include this domain knowledge into your model? For example, many of the amino acids, they're not independent, right?
Yeah, that's right.
Some of them are very similar, some are very different.
Yes, yes.
So can you put this into the model somehow?
Yeah, thanks for the question. So, okay, so you would say, for instance, incorporating the chemical knowledge of the, like, some amino acids which are hydrophobic or this kind of things, or maybe even local environments. It's true that here the hope is, I mean, in protein language models, the hope is rather that the model is going to somehow learn that by itself, right? Which is a kind of one given approach. But then you can also specialize these models. You can use them as, like foundation models and fine-tune them on a given task. And then you can give it labeled data for a given, like, if you want to predict enzyme activity, for instance, you can use measurements for that, right? It's not exactly what you asked, I guess, because you were asking rather about, can we input them at the beginning, right? Yeah, I think it would be an interesting perspective. In fact, something that I'm kind of thinking about is like the incorporation of structure, because all these methods are really fully based on sequences, but now with AlphaFold, we also have lots of structural data. And there are actually some interesting protein language models which are already partly aware of structure. And that's super interesting. And I think it kind of goes one step in your direction. Yeah.
Take a question from online. That's Sophia Anderson. She's asking about this MSA transformer that they're using in this generative way by masking and filling,
Yes.
and then converges. She's asking what would the practical use case be for such a construction. It might be interesting from a theoretical point of view, but how would you?
No, yeah, thanks. I mean, that's also a very interesting point. So in some sense, this way of generating sequences is in the family of the methods which use evolutionary data to generate new sequences, very much like Potts model, right? You rely on homologs which are known to generate new examples from this distribution. And the advantage is that you start with a given distribution. The drawback is that you cannot do de novo design, in the sense that you will not obtain a new function out of it because you're generating within a family, right? So what you can do is generate new homologs, and potentially you can then test their activity. Like, for instance, if it's in a family of enzymes, you can test their activity and then select those which have a higher activity among these proposed homologs. It's somehow it's a bit in between the traditional protein design and directed evolution, which is something a bit locally. This one is exploiting the homologs that exist. So it's very much based on evolution. And again, like, I'm not claiming to do de novo design at all with this. It's like it's within the family, but there is the hope to improve on a given function by selecting the proper protein. Yes.
Well, you could very easily turn it into like an optimization model, right? If you concatenate the specific property you want to optimize for
Yes.
in front of a sequence, and then just mask everything else and tell the model to fill it in.
Yeah. And also, I mean, with ProtMamba, we also have this thing where we want to do in-painting with the context.
A lot of questions online. I'll try to take another one here. So is there any information that the fields carry? So when you estimate a Potts model in terms of coevolutionary information, that could be interesting. Yeah, that's also an interesting point. So I mean, like in the Potts model, we do have like these one body terms and these two body terms, which, sorry, I showed. Okay, here, right. So the two body terms usually tell you about this coevolution signal between sites, and the one body terms carry information about conservation. For instance, if you have a site where it's very important to have histidine, right, then you will expect that the field tells you about this. Yeah. So the field is also important, but it's not really telling you about structure.
You have to use this information for the masking strategy. So when you mask the individual part of the sequence, you can say, okay, this part is one that I really want this to be predicted correctly. So that you can design a training protocol where you first show these bits as a missing pieces, so that the algorithm learns it faster, and then maybe this helps converging to a solution.
I think it's an interesting perspective. Yes, so far, I mean, like, you know, the masking strategy is to randomly mask 10%, for instance, of the amino acid. But yeah, I think that specifically masking some parts could be useful. In ProtMamba, with the fill in the middle, we can actually mask specifically some things. So far, we didn't really find a specific application for this, but, you know, like, we hope that, you know, there can be some interesting application. If you have any suggestions, I'm happy to.
Yeah.
I have a question about so it was very interesting that you saw when you were using this synchron optimization to pair sequences together, right, to find interactions between them. So, but you could also think about it in the other way, because there's also databases that show which proteins interact with each other, where there's empirical information, right? So you could, in principle, in cases where you don't have a lot of homologous sequences, you could pull out evolutionary information indirectly by coupling it to potential interaction partners. So
Yeah.
do you think this is a strategy to kind of expand the data sets, like by using this, or do you think that this synchron optimization is still a bit too, even if it's better than combinatorial optimization,
Yeah.
fairly expensive to do?
That's a super interesting perspective. I mean, I would say, okay, so far, I mean, okay, DishParm is a bit heavy, but okay, also because we need to use MSA transformer each time. We actually have also now a preprint on another synchron optimization, which is applied to mutual information. So much simpler, like, you know, it doesn't use a language model. It just optimizes mutual information using this differentiable trick. And this one is scalable. I mean, it's much faster. So maybe we could think of something like that. Yeah, I mean, also, okay, another thing is that you could also try to learn from examples of known complexes in order to transfer it to others. And that's something, I mean, there is some work about this by Alex Hawkins-Hooker. I mean, he looked at like some inter-domain contact, which are known to, like, you know, they're not known to interact because they are in the same molecule, right? Like, it's a multi-domain protein. And by fine-tuning a model on these, he was able to get some predictions from it. So that's also another strategy. Yeah.
One final question, I think, will be also from Sophia. And so he's asking again, isn't the concatenation of homologs quite similar to doing an MSA? I mean, the same information is there, or there's some difference, right? But maybe you can comment.
Yeah, so I mean, it is somewhat similar. So really, the hope is to try to isolate the two notions of homology on the one side and alignments on the other side. Like, we want to use homology because it seems that it's very useful for models. Even I showed you ESM2 is clearly learning about homology in the way it's capturing the contacts. Sorry, I guess it's like, okay, it's here. Yeah, so like homology seems to be really important. But alignments, again, they have these imperfections. They are also slow to calculate and so on. So by concatenating, we are doing something which is similar to alignments for sure, but we are not requiring the step of alignment, which can have mistakes by, you know, like the gap can be put in the wrong place, right? I mean, this stuff like that. So we avoid this. The sequences are just the sequences as they are, and concatenating them means that the input is structured in families of homologs, but without having, we hope, without having the defects of alignments.
Don't have the exact metric, sort of, don't have a proxy for the similarities.
Yes, exactly. I mean, like, we are not feeding the model with the knowledge of, you know, which column of what sequence correspond to which column of the other one. It's just seeing a set of homologs. So it is similar, but the hope is just to get rid of the alignment issues. Yeah.
Okay. Thank you, Anne-Florence, for a wonderful talk.
Thank you very much, Sean.